Welcome to Linea Light Podcast Section. Visit our website linealight.com.
Lighting Talk with Dr. Michelle Maclean. Hins Light, the environmental decontamination that protects people.
Good morning everyone and welcome back to our appointment with light. This episode introduces the scientific research behind the new Hins technology, which is all about bacterial decontamination. It is with pleasure that I welcome our guest today, Dr. Michelle Maclean, from the University of Strathclyde's Department of Electronic and Electrical Engineering.
Good morning, Sylvia. Thank you for the introduction and for inviting me along. It's a pleasure to be speaking to you all from here in Scotland.
So we are talking about something really innovative. Would you like to explain what is meant by Hins Light and what is the discovery your team has made?
So, Hins Light is the abbreviation for high intensity narrow spectrum light, and this is an antimicrobial technology which uses visible light in the violet-blue region with peak activity around 405 nanometer wavelength. And our research at Strathclyde has focused over the years on establishing the antimicrobial effects of 405 nanometer light and also its potential for infection control applications. And a major area of our work has been the development of this technology for environmental decontamination. Our work's demonstrated not only the broad antimicrobial efficacy of these light wavelengths, but that these antimicrobial effects could be achieved at levels that are safe for human exposure. And this enabled us to develop a visible light system that is safe for continuous use in occupied and unoccupied environments. Now, this technology, which we termed the Hins Light Environmental Decontamination System, or EDS for short, is a ceiling-mounted lighting system that is designed to be used continuously, in the same way as a standard lighting system, but with the ability to continuously decontaminate the air and exposed surfaces around the room, essentially providing a whole room decontamination treatment. Now, the EDS is designed to be used in combination with existing cleaning and infection control procedures, and clinical evaluation of the technology has demonstrated that a significantly cleaner environment can be achieved when using the Hins Light technology.
For your trial, you initially chose a specific ward in the Glasgow Royal Infirmary, the burns unit, as the test area. Why this choice?
Yes, so we've conducted the majority of our clinical evaluations in the Glasgow Royal Infirmary, which is a major teaching hospital located near to the University of Strathclyde campus. These clinical evaluations involved close collaboration with infection control experts and clinical and nursing staff at the hospital, and they were carefully set up to evaluate the effectiveness of the Hins Light EDS at various locations, including the burns unit, as you mentioned, and also the intensive care unit. The technology itself could be utilised in any area within hospitals. However, there are areas that can be specifically targeted, such as burns units, intensive care units and operating theatres, for example, where there are either patients that are highly susceptible to infection or areas where there is an increased risk of infection. So, in discussion with clinical colleagues, this is why we chose to conduct our initial evaluations in these key areas.
Can you just explain in more detail how you carried out the trial?
The majority of our studies have been conducted in isolation rooms or clinic rooms, and we collaborated closely with clinical colleagues and statisticians to ensure that the study design was carefully planned. For evaluation, the Hins Light decontamination system, or systems depending on the size of the clinical area, were installed in the ceiling of the isolation room or clinic being treated, and then the efficacy of the system was evaluated by monitoring the levels of bacterial contamination around the room before, during and after use of the system. Now, this is what's known as a before and after intervention study design, where measurements are made before and after the application of an intervention, which in this case was the use of the Hins Light system. And this allows the effects of this intervention to be evaluated. So to conduct the studies, we measured the levels of bacterial contamination present on a range of contact surfaces around the room. And we did this using contact agar plates, which allowed us to measure both the total bacterial contamination but also the levels of staphylococcal bacteria. And these are organisms that provide a good representation of contamination from human sources. We measured the bacterial levels for a period before the Hins Light EDS was switched on, and then repeated these measurements after the system had been in use for a period, and then again for a period after the light system was switched off. And typically 50 to 100 samples were collected during each phase of the study, and the duration of each study varied from 3 days to 3 weeks, depending on the clinical situation. During the studies, rooms were occupied by a patient, and normal cleaning and infection control procedures were carried out so that the bacterial reductions obtained were over and above those achieved by the normal cleaning and infection control procedures.
There are other decontamination techniques and technologies. So why should I choose Hins technology instead of ultraviolet light or hydrogen peroxide vapor?
So the Hins Light technology is different to the other technologies you mentioned. Ultraviolet light and hydrogen peroxide vapor are very effective for room decontamination, but they're what are termed terminal clean technologies. These technologies are used to decontaminate a room or area after it's been vacated, and rooms need to be empty and sealed off before HPV or UV treatment is applied because they're not safe for use in occupied areas. The Hins Light technology is different in that it is used to provide continuous decontamination of occupied environments. And the purpose of this is that use of the technology provides a gentle, continuous decontamination treatment, which prevents the environmental contamination around the room, in the air and on contact surfaces, rising to the high levels that would be typical of occupied rooms and areas. And being able to maintain low levels of environmental contamination means a cleaner environment and a reduced risk of infection arising from environmental sources.
Dr. Maclean, this could be the beginning of a new era, the era in which luminaires are not only functional and high-performing from a lighting point of view, but provide benefits to people by helping make environments safer. I'm very pleased to have had you as a guest today and to have been able to present your innovation in the area of scientific studies, so thank you very much again for joining us.
Thank you very much, Sylvia. It's been a pleasure speaking to you all today, and I hope everyone has enjoyed hearing more about the Hins Light technology.
Thank you for joining us. Discover more on our website linealight.com.